Next discussion, anatomy. Under anatomy, T.S of Dicot and Monocot root both. So both Dicot and Monocot root, both roots shows three parts: epidermis, cortex and stele. Even in Monocot root, it shows three parts: epidermis, cortex and stele. So first one, epidermis. Epidermis is single layered, made up of tubular or rectangular cells. They contains vacuolated protoplast, living cells. Then the cells contains leucoplast, colorless. Then in a root, that epidermis is without stomata, cuticle, both are absent. Even intercellular space is absent. Some of the epidermal cells smaller. The smaller cells, their cell walls get extended. Such extensions of that epidermis called root hairs. Then cells developing root hairs are called trichoblast. Cells developing root hairs are called trichoblast. Because of root hairs, root epidermis is called 𝗹𝗶𝗸𝗁 or 𝗘𝗍𝗺𝗺 layer or rhizodermis. This is epidermis of dicot root. Then epidermis of monocot root. Same characters. When monocot root, epidermis is unilayered, made up of tubular or rectangular cells, contains vacuolated protoplast, even leucoplast. The smaller epidermal cells produce extensions. These extensions are called root hairs. Then cells developing root hairs called trichoblast. Due to root epidermis, due to root hairs, root epidermis is called 𝗹𝗶𝗸𝗁 or 𝗘𝗍𝗺𝗺 layer or rhizodermis. So dicot, monocot root, same characters. One exceptional only in epiphytes. In epiphytes, root epidermis is multi-layered, dead, hygroscopic, rightly called velamin tissue. That is exceptional only in one family. So generally epidermis is same. Next one, cortex. Cortex. Root character. Cortex is enlarged. Cortex enlarged. And this cortex shows three parts: exodermis, then general cortex and dermis. Then first one, ectodermis. So cells below the epidermis, initially they are parenchymatous. Later, due to deposition of suberin, they become thick-walled, called exodermis. In dicot root, exodermis is 2 to 3 layered. Then, function of exodermis. If epidermis is ruptured, it means old root. In old root, if epidermis is ruptured, this exodermis acts as protective layer. And second one, because of suberin, it prevents leakage of absorbed water. That is exodermis. Even monocot root, that cortex shows 3 parts: exodermis, then general cortex, and endodermis. Same even in monocot root, outer cortical cells, due to deposition of suberin, they become thick-walled. In old root, they act as protective layer, and they prevents leakage of absorbed water. This is exodermis. Next one, general cortex. General cortex present in between exodermis and endodermis. Exo and endodermis. And general cortex means they are parenchymatous. So parenchyma means cells oval or spherical or round, loosely arranged. So intercellular spaces present. Then function of general cortex. One is help in lateral conduction. And these cortical cells, they contains leucoplast, so help in storage. Then point is only in photosynthetic roots. This general cortex contains chloroplast, so help in assimilation. Then general cortex in monocot root. Same. Even in monocot root, general cortex present in between exodermis and also endodermis. And here also cells parenchymatous, so oval or spherical in outline, then intercellular spaces present, they contains leucoplast, help in storage, and in both roots, along with storage, they help in lateral conduction, that too mostly through cell wall and intercellular spaces, rightly called apoplastic pathway. And here in monocots, in vellum epiphytes. In epiphytes, epidermis is dead tissue, and then in teniophyllum is epiphyte, but it has photosynthetic roots. So epidermis is dead tissue. So in teniophyllum, this general cortex contains chloroplast, so help in assimilation. Now, next one, endodermis. So endodermis, innermost layer of cortex is called endodermis. In roots, endodermis is well developed. Generally, endodermis unilayered, made up of barrel-shaped cells and cells closely arranged. So, without any intercellular spaces. Without any intercellular spaces. Generally, this endodermis, it shows two types of cells. Among two types, some of the endodermal cells, they show tangential and also radial thickenings. These are tangential and these are radial thickenings. These thickenings are due to deposition of lignin and suberin. Such thickenings are called Casparian thickenings, reported by Caspary. Because of Casparian thickenings, this endodermis act as barrier between stele and the cortex. Because of these thickenings, it is impermeable to water. So, some of the endodermal cells exactly opposite to protoxylem are thin-walled. Such thin-walled cells are called passage cells or transfusion cells. Through that, conduction takes place. Same in monocot root. Even in monocot root, innermost layer is called endodermis. Same characters. Even in monocot root, that endodermis unilayered, made up of barrel-shaped cells. Then some of the endodermal cells shows a radial and a tangential thickenings called Casparian thickenings. They are impermeable to water, or because of that, endodermis act as barrier between cortex and stele. Then some of the endodermal cells opposite to protoxylem are thin-walled, called passage cells or transfusion cells through which conduction takes place. This is epidermis and cortex. Up to here, almost dicot and monocot root shows common characters. Now, next one is stele. Next one is stele. Stele shows pericycle, medulla, then conjunctive tissue and vascular bundles. So, first one, pericycle. Generally, pericycle is unilayered. A rare case, it is multi-layered. So, pericycle is outermost layer of stele, uni or multi-layered and parenchymatous. In dicot, monocot root, pericycle outermost layer, uni or multi-layered, then by differentiation this pericycle produce lateral roots. So, both in dicot and monocot root, common function that pericycle produce lateral roots. Now, first difference, in dicot root, even after maturity, this pericycle is parenchymatous and by dedifferentiation it produce cambium. Produce cambium means produce both cork cambium and vascular cambium. So, in dicot root, secondary growth takes place. But in monocot root, old monocot root, in old monocot root, due to deposition of lignin, this pericycle becomes sclerenchymatous. So, it does not show any differentiation, dedifferentiation. So, cambium is not formed. So, secondary growth is absent. This is pericycle. Then, next one, vascular bundles. Vascular bundles, it includes xylem and phloem. Xylem and phloem. Then point, this xylem and phloem separate, alternate, then present on different radii called radial vascular bundles. So, root character, root either dicot or monocot, root character, so radial vascular bundles. Radial vascular bundles. Then, xylem differentiation, centripetal, in which protoxylem towards outside, means towards pericycle. Metaxylem towards medulla. So, that centripetal differentiation gives exarch. So, root character is vascular bundles are radial. means vascular bundles radial means xylem and phloem. Separate, alternate, present on different radii, called radial vascular bundles. Even one more root character, that xylem differentiation is centripetal. Xylem differentiation centripetal means this protoxylem outside, metaxylem inside, called exarch. Called what? Exarch. Now, difference is in Dicot root, limited vascular bundles present. Dicot root, limited vascular bundles present. Common is tetrarch. Only 4 xylem and phloem bundles present. But some exceptional, only one vascular bundle in Dicot root. Common is tetrarch, but some exceptional, only one vascular bundle, monarch. Example, trappa. Diarch, 2 vascular bundles. Example, nicotiana. Triarch, 3 vascular bundles. Example, pisum. Tetrarch, 4 vascular bundles. Example, gossypium, cotton. Pentarch, 5 vascular bundles. Example, ricinus, castor. Octarch, 8 vascular bundles. Example, castanea. These are exceptional in dicots, but most common is tetrarch. But here it is triarch, 3 vascular bundles. Then monocot root. In monocot root, many vascular bundles present. Like monocot root, many vascular bundles present. That is called polyarch. So many vascular bundles called polyarch seen in monocot root. Example is maize. Then next one, medulla. In dicot root, medulla is reduced or scanty because during xylem differentiation, this medulla reaches to center. Then monocot root. In monocot root, medulla is enlarged. Enlarged medulla, monocot root, and it is parenchymatous in young root. Then next, root character. This vascular bundles, they are connected by parenchymatous cells. These parenchymatous cells are called conjunctive tissue. Parenchymatous cells called conjunctive tissue. Called conjunctive tissue. This conjunctive tissue present in both dicot and also monocot root conjunctive tissue. Point is, in dicot root, this conjunctive tissue, dicot root, conjunctive tissues parenchymatous help in storage and even by dedifferentiation it produce cambium. In old monocot root, conjunctive tissue is a sclerenchymatous. So it provide only mechanical support. So now differences between dicot and monocot root. First one, ectodermis. It is 2 to 3 layered in dicot root. Monocot root, it is 1 to 2 layered. Cortex is extensive in monocot root when compared to dicot root. Then in dicot root, pericycle by dedifferentiation produces cork and vascular cambium. So at dicot root, secondary growth takes place. Monocot root, matured monocot root, pericycle is sclerenchymatous. So it does not undergo any dedifferentiation. So no cambium formation. So no secondary growth. Then dicot root, medulla is reduced. Monocot root, medulla is enlarged. In dicot root, limited vascular bundles present. Common is tetrarch. Here monocot root, numerous vascular bundles present. That is called polyarch. Then in dicot root, conjunctive tissue by dedifferentiation produces vascular and cork cambium. But monocot root, matured monocot root, conjunctive tissue is sclerenchymatous. So these are about dicot and monocot root. T.S of dicot stem and T.S of monocot stem. So first one, T.S of dicot stem. T.S of dicot stem, it shows 3 parts. One is epidermis. Then second one, cortex, and third one is a stele. So it shows 3 parts: epidermis, cortex, and a stele. So first one, epidermis. Epidermis is outermost layer, then single layered, made up of rectangular cells. That is, cells are closely arranged. So intercellular spaces are absent, and cells are living cells. They contains vacuolated protoplast, but a chloroplast is absent. Then the stem epidermis, it contains stomata and cuticle. So, function of stomata. Through the stomata, transpiration and even exchange of gases, respiration takes place. Then this cutin, actually epidermal cells secrete cutin. This cutin forms layer, rightly called cuticle, and cuticle function is it reduces transpiration. Then this epidermis also produce multicellular outgrowths. Rightly, these multicellular outgrowths are called trichomes. Trichomes, multicellular, then uniseriate, that cells arranged in one row, or biseriate, cells arranged in two rows, or multiseriate, cells arranged in many rows. Then function of trichomes. These trichomes also reduce that transpiration rate. Then trichomes also prevents that entry of pathogens. This is about epidermis. The next one is cortex. Cortex, actually cortex shows three parts. One is outer cortex. Outer cortex is called hypodermis. Then middle cortex is called general cortex. Third layer, endodermis. So point is, cortex differentiated into three layers or three parts: hypodermis, general cortex, then endodermis. Hypodermis, it is present below the epidermis. Hypodermis. Generally, hypodermis is several layered, and particularly in dicot stem, this hypodermis is formed by 𝗂𝗽𝗱𝗁. Hypodermis is formed by 𝗂𝗽𝗁. Then in helianthus, sunflower, this 𝗂𝗽𝗱𝗁 hypodermis is homogeneous. It forms continuous layer. And in cucurbita, it is opposite. In cucurbita, 𝗂𝗽𝗱𝗁 hypodermis, it forms a discontinuous layer. Discontinuous layer. Then rightly 𝗂𝗽𝗱𝗁 is living tissue. Living tissue, more amount of pectin is present. Then right, it is called a pectinized mechanical tissue. Why means it provides mechanical support. Particularly, it gives tensile strength or elasticity. It gives tensile strength or elasticity. Then this living tissue, so in young stem, it contains chloroplast. In young stem, it contains chloroplast, so it helps in photosynthesis or assimilation. So function of 𝗂𝗽𝗱𝗁 hypodermis, two. One is it gives elasticity or tensile strength, and second one, photosynthesis or assimilation. This is hypodermis. Then middle cortex, middle portion or middle cortex is called general cortex. General cortex is predominantly parenchymatous. So it is made up of parenchyma. So that cells a spherical oval with intercellular spaces, generally intercellular spaces present. And outer cortical cells contains chloroplast. Outer cortical cells contains chloroplast, so help in assimilation. And inner cortical cells contains leucoplast, so they help in storage. That is about cortex, general cortex. Then next one, endodermis. So this is endodermis. Endodermis, that is innermost layer of the cortex. Endodermis is generally unilayered and cells made up of ligno-suberin thickenings are called Casparian thickenings. Then intercellular space is absent. Then here in Dicot stem, that endodermis stores starch, hence this endodermis is called starch sheath. Here one question, photosynthetic parts in that Dicot stem is, one is hypodermis and outer cortical cells. They perform photosynthesis, not endodermis. Endodermis simple, it stores starch, but it does not produce starch. So point is, photosynthetic parts means hypodermis and outer cortical cells. And which one stores starch means endodermis. This is about epidermis and a cortex. The next one, stele. Stele actually in Dicot stem. In Dicot stem, stele is called a eustele. Dicot stem, that stele is called eustele. Eustele means ring of vascular bundles present. If vascular bundles arranged in a ring, that is called eustele. Dicot stem, limited number of vascular bundles present. Generally 10 to 20 vascular bundles present, but all that vascular bundles arranged in a ring, in the form of ring, so such stele is called eustele. Then these parts of stele, generally stele shows 4 parts. Among these 4, one is pericycle. Then second one is a phloem, vascular bundles, pericycle, then vascular bundles, medulla and medullary rays. These are parts of stele. One is pericycle, second one is vascular bundle, third one medulla and medullary rays. So first one, pericycle. Then in Dicot stem, that pericycle is multi-layered, generally multi-layered and sclerenchymatous. Here in helianthus, in sunflower, this sclerenchymatous pericycle occurs as a patches. It means not uniform, discontinuous. It occurs as patches, and that patches of sclerenchymatous pericycle is called bundle cap. Bundle cap. Actually, in between bundle caps, there is parenchymus present. It means in sunflower, hypodermis forms continuous layer, but in sunflower, pericycle is discontinuous, rightly called bundle cap, and this pericycle surrounds that phloem and provide mechanical support. So it is also called, sclerenchymatous pericycle is also called hard bast. So, point pericycle, multi-layered, sclerenchymatous. In sunflower, this sclerenchymatous pericycle contains patches of parenchyma and forms bundle cap or that hard bast. In cucurbita, opposite. In cucurbita, this hypodermis forms discontinuous, but this sclerenchymatous pericycle forms continuous layer in cucurbita. This is about that pericycle. Next one, vascular bundles. Vascular bundles, rightly in dicot stem, vascular bundles are wedge shaped. Wedge means that top shaped. And among vascular bundles, generally phloem is outside. Among vascular bundles, phloem is outside. Then xylem, the xylem is inside. But phloem and xylem present on same radius. Phloem and xylem present on same radius, called collateral vascular bundles. Generally, these vascular bundles are joined, called conjoint. But between xylem and phloem, there is cambium. So, it is called open type. So, point is once again that vascular bundles arranged in the form of ring called eustele, and in vascular bundles phloem is outside, xylem is inside, both are present on same radius called collateral, both are joined called conjoint. Between xylem and phloem there is cambium, so it is called open. Your point, vascular tissues two types, primary and secondary vascular tissues. Primary vascular tissues, they develop from procambium. Primary vascular tissues develop from procambium. After that, some amount of procambium persists in between phloem and xylem, and such cambium is called intrafascicular or fascicular cambium, and it is primary. This is about vascular bundles. Then in vascular bundles, phloem, this is phloem and a xylem. Right, it is dicot stem. So, in dicot stem, that phloem contains sieve tubes and companion cells, even phloem parenchyma. Generally, secondary phloem contains phloem fibers, absent in that primary phloem. In the same way, xylem, this xylem shows tracheids, vessels, even xylem parenchyma and xylem fibers. Then point, primary xylem actually two types. Primary xylem is two types. Newly formed xylem is called protoxylem. So, it is protoxylem. Newly formed xylem is called protoxylem. Later formed xylem is called metaxylem. Metaxylem. It means in stems, xylem differentiation takes place from center to periphery. So, at center, it means towards medulla, protoxylem is present. And outside means towards pericycle, metaxylem is present. Such condition is called end arc. So, stems, xylem differentiation is centrifugal. So, that xylem is end arc. These are vascular bundles. So, pericycle, vascular bundles. The next one, medulla. Your point is, in dicot stem, ring of vascular bundles present. So, at center, enlarged medulla is present. Center, enlarged medulla is present in dicot stem. These medullary cells, rightly parenchymatous, and they help in storage. One exceptional, 𝗂𝗽𝗱𝗁𝗃, dicot family, 𝗂𝗽𝗱𝗃. In 𝗂𝗽𝗱𝗃, this medullary cells degenerates and forms that medullary cavity. Then some of the medullary cells, they extend in between vascular bundles. Some of the medullary cells, see here, some of the medullary cells, they extend in between vascular bundles and forms medullary rays. This medullary rays present only in dicot stem and gymnosperms. Then point, what is the function of medullary rays? Generally medullary rays parenchymatous. This medullary rays for radial conduction. They help in radial conduction. And at the time of secondary growth, these medullary cells by dedifferentiation produces interfascicular cambium. So, point is, there are two types of cambia in dicot stem. One is intrafascicular, that is primary meristem, and intrafascicular present between primary phloem and xylem, called intrafascicular, which is persists part of that procambium. Second one, interfascicular. Interfascicular means it is present in between vascular bundles. Interfascicular, present in between vascular bundles, called interfascicular. That interfascicular cambium is formed from medullary rays by dedifferentiation, so it is secondary. So, point is, one is intrafascicular, which is primary meristem. Second one, interfascicular, which is secondary meristem. This intra and interfascicular cambium joins to form vascular cambium. So, this is structure of dicot stem. Next one, T.S of Monocot stem. T.S of Monocot stem also shows three parts. One is epidermis, cortex, and third one stele. So, first one, epidermis, almost same characters. Epidermis, outermost layer, single layered, protective in function. Then this epidermis is made up of that rectangular cells, that cells closely arranged. Cells contains vacuolated protoplast. So, cells are living cells, but no chloroplast, without a chloroplast. Then this epidermis is surrounded by that cuticle, then it also contains stomata. So, function of stomata, help in respiration and also transpiration. Then function of cuticle, that cuticle reduce that transpiration. Then in monocot stem, that epidermis is without trichomes. So, trichomes are absent. Only in dicot stem, the trichomes present. That is first difference. The next one, cortex. In monocot stem, cortex is highly reduced. Cortex is highly reduced. This cortex is represented by only hypodermis. Cortex is represented by only hypodermis. But in monocot stem, this hypodermis is made up of sclerenchyma. In dicot stem, it is made up of collenchyma. In monocot stem, it is made up of sclerenchyma. So, sclerenchymatous hypodermis means dead tissue. So, dead tissue, so it provides only mechanical support. Then, bulk portion of monocot stem. Bulk portion of monocot stem is that a ground tissue. It is ground tissue. This ground tissue is made up of parenchymatous cells. It is made up of parenchymatous cells. Ground tissue, it is made up of parenchymatous cells. Then, the outer cells. These outer cells are larger, and outer cells contains chloroplast. So, help in photosynthesis. Inner cells are smaller. Inner cells smaller. They contains leucoplast. So, they help in storage. Then, monocot stem. In monocot stem, that stele is called attactive stele. Attactive stele, that scattered vascular bundles present. So, in monocot stem, vascular bundles scattered. So, such stele is called attactive stele, and attactive stele is advanced type. Stele includes only vascular bundles because pericycle, medulla, and even medullary rays are absent. Rightly, vascular bundles are called fibrovascular bundles. So, vascular bundles are called fibrovascular bundles because these vascular bundles are surrounded by sclerenchymatous sheath. So, called fibrovascular bundles. Then, even in this, that a phloem is outside and a xylem is inside. So, phloem and xylem both are present on same radius. So, it is called collateral vascular bundles. Then, phloem and xylem is joined, called a conjoint. But here the joining is direct because no cambium. So, rightly called closed type. So, in monocot stem, vascular bundles are collateral, conjoint, and closed. Among xylem, in monocot stem, the xylem contains few xylem vessels. Generally, 2 metaxylem and 2 protoxylem vessels present. But protoxylem vessels present. Some of the protoxylem vessels, they degenerates and forms cavity. That cavity is called protoxylem lacuna, and it stores water. So, it is called water storage cavity. So, monocot stem contains protoxylem lacuna. Rightly, this protoxylem lacuna is lysogenous cavity. It is formed due to that lysis of cells. Due to lysis of cells. Generally, xylem shows tracheids, vessels, that xylem, parenchyma, and fibers. Phloem shows sieve tubes, companion cells, and which one is absent in monocot stem means that a phloem parenchyma is absent. Then, vascular bundles are scattered. That is why medulla, even medullary rays are absent. It is about monocot stem. Now, differences between dicot and monocot stem. Dicot stem, trichomes present. Monocot stem, that the trichomes are absent. Then, second one, cortex, dicot stem cortex is comparatively well developed with a root, with stem, monocot stem. So, hypodermis, that hypodermis is 𝗂𝗽𝗱𝗁𝗪𝗔 and dicot stem. So, it performs two functions, elasticity and also photosynthesis. But in the monocot stem, that hypodermis is 𝗂𝗽𝗱𝗁𝗪𝗔. So, it gives only mechanical support. Then middle cortex, that is called general cortex. General cortex, outer cortical cells perform photosynthesis, inner cortex storage, but here general cortex is absent. Then endodermis, innermost layer of cortex called endodermis. In dicot stem, endodermis stores starch, so it is called starch sheath. Monocot stem, starch sheath is or endodermis is absent. In dicot stem, pericycle is multilayered, sclerenchymatous, but in sunflower, along with sclerenchyma, it contains parenchyma. In monocot stem, pericycle is absent. Then, dicot stem, that stele is eustele. Eustele means a ring of vascular bundles present, primitive. Monocot stem, that stele is adactostele, scattered vascular bundles present. Then, in dicot stem, vascular bundles generally wedge or top shaped. Monocot stem, this vascular bundles oval in outline. Then, in dicot stem, vascular bundles are open, means between them cambium is present, rightly called collateral conjoint open. Monocot stem, vascular bundles are closed, means collateral, conjoint and closed. In monocot stem, this bundle sheath is surrounded by sclerenchyma, so rightly called fibrovascular bundles, but in dicot stem, normal vascular bundles present. Then, in monocot stem, phloem is without phloem parenchyma. In dicot stem, there is phloem parenchyma. In monocot stem, there is protoxylem lacuna. It is lysogenous cavity. In dicot stem, such cavity is absent, but only in 𝗂𝗽𝗱𝗁𝗪𝗔 there is medullary cavity. Dicot stem, there is enlarged medulla. In monocot stem, medulla is absent. Then dicot stem contains medullary rays for lateral conduction and also participate in secondary growth by producing interfascicular cambium. So, in monocot stem, general cortex, endodermis, pericycle, medulla, medullary rays all are absent. And in monocot stem, bulk portion of monocot stem is formed by that ground tissue. And in monocot stem, cortex is highly reduced, is represented only by hypodermis. So, these are differences between dicot and monocot stem.